good afternoon to all of you. And first, before starting this introduction or this general presentation, I just have to tell you that please interrupt me if you don't follow correctly or if you have any questions during the talk. Let's make this informal and useful for everyone. So I am introducing this introductory slide to my presentation just to place this talk in a more general context within the framework of this astrobiology training course. And you have heard during these days, and you will in the next few days, that astrobiology deals with the origin and evolution of life in the universe, and that in a context of natural sciences, this implies that there is prebiotic chemistries and kind of compounds that at a given point get together, somehow interact to form the first life forms. And how to study those first life forms is very difficult because it's a past event and it is in part contingent, historical. So we can do that, and we try to do that using multiple approaches, and you have heard already about this, bottom-up approaches, and you have heard more about this type of approaches where you analyze or study how atoms form in our universe and how molecules and increasingly complex atoms form. And then little by little you can sort of come up to a kind of prebiotic world where things can happen. The other approach that is quite complementary is a top-down approach that consists on saying, let's take one example of life, and the only one that we know is life on Earth, and let's try to identify things that are characteristic of life and that kind defines some sort of common denominator, and from this approach we can perhaps hypothesize about how the first life forms were and how they came to be. So this is what biologists can do. And of course, there are many other disciplines involved in these two types of approaches to the study of the origin of life. So my talk today is going to be on this far right end of the slide. So I'm going to talk about biodiversity today on Earth. So I'm a biologist, and this is what I'm going to do. I will talk about the extent of life on Earth and its limits. And perhaps this can be useful to understand how past forms evolved and also what are the limits of life, maybe in the material universe that we know. So there will be two parts in my talk. One more general, in which I will introduce biodiversity and especially microbial biodiversity, because as you will learn, most biological diversity belongs to the microbial world. And we will focus a little bit on the different techniques and methods that we can use to explore the diversity that escapes to our human eye. And then in the second part, I will talk about the limits or the physicochemical limits that we know experimentally on our planet. So this is it. And of course, humans have classified life for many centuries into different types of organisms and have given species names, etc., essentially of animals and plants. So most very old taxonomic schemes concentrate on animals and plants. But we know now that most biological diversity, as I said, is microbial. And this we know now, but it has been a long run, or it has taken a long time to realize that. And this is because we have an observational bias. First, because of physical constraints. We are big organisms, and we can identify or differentiate animals that are our size better than other. There is also an anthropocentric bias that is quite universal in general. So each individual places itself in the center of the universe, and it is very normal that humans do so as well. And then, but these observational biases and this normal anthropocentrism biases or interpretation of the natural world. And here I have to point out or highlight some false ideas that we have. And one is that evolution necessarily means an increase in progress and complexity. This is not necessarily the case, biological evolution. And we have many examples of this. So you can have, for instance, evolution by reduction and loss of complex characters. And I want also to warn you about the risk of identifying simple as primitive. It is not the same. So you can have simple things that are highly adapted things, or imagine many parasites, for instance, have reduced morphologics and structures and genomes just because they are adapted to make a very specific function. So you can have simple organisms that are fit to their particular environment and requirements, and they are by no means primitive. So these are false ideas. I just want to warn you against these generally taken ideas, which are not true. So an example of how we bias and the problem of understanding and studying microbes is, as I say, our perception of biological diversity is biased. If you see to those all these faces, you can recognize, especially the French, many of these individuals and put names under them, under the pictures. But we Occidental Western Europe individuals have already more problems to identify and put names under faces of more distant populations, human populations. And the opposite is also true. Okay, so this has been studied for a long time. But when we go to more distant phylogenetic species, other species, like flies, for instance, you start to get even more problems to recognize individual organisms. And when you go further to a much more distant, at a farther evolutionary scale, you get to a point that you can not only, you are unable not only to identify or differentiate individual organisms by their phenotypical features, by their features, but you are unable to distinguish species, microbial species among them. Okay, this is not only this is in part because of the phylogenetic distance, the evolutionary distance. So things that are more evolutionary distant from ourselves are more difficult for us to differentiate. It is also due to the fact to a spatial scale, so it's a problem of scale, because microbial species are small, they tend to have less appreciable characters for us, which are much bigger organisms, so we have more problems to recognize individual differences in this type of organisms. And I have put also this temporal scale because of another problem that we humans have that has to do with our capacity to really grasp long temporal scales and time and evolutionary time. This is precisely more explained in this slide. It means that we don't know exactly when life arrived, emerged on Earth or evolved on Earth, somewhere between 3.5 and 4 point something billion years ago. And we know that for a long period on the evolution of life on Earth, life was essentially microbial. Large multicellular organisms such as animals and plants date back as perhaps 500 million years, but life arose much earlier. So this implies that there was a very long evolutionary time where life was essentially microbial. Mostly prokaryotic, bacteria and archaea, but also microbial eukaryotes appear at some time. So this implies that there was a huge evolutionary time before large organisms that have complex tissues emerge. And this is also an opportunity for evolution to take place. And this explains something that we see today in biology, and is that most biological and genetic diversity belongs into the microbial world. And this is all in large part due to the fact that microbial organisms have had a much long evolutionary time. So they have diverged much more because there is a much longer temporal scale that you need to take into account. So this is something that we often forget because simply we have difficulties to grasp billions of years. It's for us something that we cannot measure easily or comprehend. So this explains in part our difficulty to deal with the microbial world. First, we as humans took a long time to discover the existence of microbial life. So this happened more or less at boundary between the 17th, 18th century with the discovery or the development of the first microscopes, quite rudimentary at the time. And then, thanks to the improvement of microscopic techniques and instruments, we little by little discovered that this microbial life was more diverse than we could think of at the beginning. And for instance, this allowed first to realize the fact that all organisms are composed of cells. We animals, but also in this type of first animalcules, as Van Leeuwenhoek called these kind of organisms that could be seen under the microscope. But then it also, this observation of microorganisms with increasingly powerful microscopy techniques led to the elaboration of the first universal phylogenetic trees based on morphology, on morphological characters, and this happened in the 19th century thanks to Ernst Haeckel, very famous evolutionist, Darwin's follower for a long time, and who for the first time tried to include all organisms in a single phylogenetic tree, in a tree where the different branching point indicated relationships of evolutionary relationships. And in this tree that he depicted based on morphology at that time, he included not only plants and animals, but he included a third group of organisms that he called protista at the time, that included what we know today as bacteria, and also many microbial eukaryotes in general. Fungi, for instance, were there, etc. So today we call protists to microbial eukaryotes. It's another different definition, but the first time that someone proposed this name, it was Ernst Haeckel for, let's say, globally unicellular life. Okay, so there was already an attempt to include microbes in a universal classification of life. And of course, well, some pictures of how these protists may look like. And then, of course, electron microscopy came, and it helped to recognize another very important thing, which is valid today, and it was quite late, by the way, 20th century, and it is the fact that all cells on our planet fall into two distinct structural categories. They can be prokaryotic, like bacteria, for instance, which have the DNA, the genetic material directly immersed within the cytoplasm, within this kind of gel where most biochemical reactions take place. And then the eukaryotes or eukaryotic cells that are apparently more complex because they have an internal endomembrane system and an internal membrane system with many different kinds of compartments, but one of them is very important. It's called the nucleus and separates physically the genetic material, the chromosomes, and the rest of the cytoplasm where many other activities, including protein synthesis, take place. So these are very important things that we discover thanks to increasingly developed methods or techniques of microscopy. But then this was relatively limited in morphology. Then came the molecular biology revolution. So during the 20th century, we discovered many things, including the fact that the genetic material in cells is composed of DNA containing that encodes for proteins later in the molecular biology steps, and then the DNA structure allowed to understand many other processes. And then for a long while in the 20th century, we learned many things about how cells work in terms of molecular biology. So we understood that cells have a dual function, and this is important because these properties are somehow universal. The details may be different, but the properties are universal. With, on one side, there is a function, a cellular function that has to do with the storage, transmission, and expression of genetic information. So you have DNA, which is the genetic material in cells, that can be passed to following generations by a process that is called replication, DNA replication, which is semi-conservative. And then you can also have the expression of the information contained in the DNA via transmission and an intermediate RNA molecule. That is, this information is translated into proteins in particular machineries, which are called ribosomes. So, and these proteins can make a variety of functions, from structural to more catalytic activities. So this is part of the activities of a cell, and the other half concerns the transformation of matter and energy, and this is made possible by a variety of proteins, but also by the presence of a membrane across which we can generate ion gradients, in particular proton gradients, which are exploited by current cells to produce free energy for a variety of functions. So this is, in essence, these are the molecular biology properties of a cell that are quite universal. And so during the second half of the 20th century, we have learned a lot about all the molecular processes that take place within a cell, and we are still learning. So there's a lot of room for progress in that. But one thing that was very difficult to tackle, and it was a problem for a long time, was to establish a natural classification for all organisms, and particularly for microbes. So we were at a point where we could study how cells work intimately within the cell, within the cell boundary, the membranes, but it was not possible for us, for microbiologists and for biologists in general, to establish a natural classification. A natural classification is a classification that is based on evolutionary relationships of vertical descent among organisms. And this was not possible because microbes, and especially in the case of prokaryotes like bacteria, there are not enough morphological differences. So it was very difficult to say who was related with whom, because there are thousands and thousands of species that look like this. So you may have some cocci or little rods, but so tiny differences, but there are many species of rods and of bacilli, this type of rod or cocci. So this means it is very difficult to differentiate macros just based on morphology. To the point that by the 1960s, a very famous microbiologist, Roger Stenier, complained and in a sense gave up and said it is impossible to achieve this ultimate scientific goal, that is to achieve a natural phylogenetic classification of life. This cannot be achieved in the case of bacteria because of what I just mentioned. Things, of course, so at the time, what microbiologists did, and did it for a long while, for many, many years, was to establish a parallel taxonomy, a parallel system of classification that was called numerical at the time, where you could study the properties of organisms that could grow in culture in the lab. So you can take, and this is how we do today, so this is classical techniques if you want. You can take your samples from soils, from seawater with this type of bottles, etc. And you can try to isolate microbial species, let's say, by successive dilution in liquid media or by plating cells onto this type of agar media, more solid media, with the idea to get clonal populations, which we call pure cultures, where the whole population derives from a single cell, ideally, and then you may imagine that cells are more or less equal, and then you can study the morphology if you want in further detail, ultrastructure if you can, but also you can study the properties of this clonal population in a variety of culture media to see whether these organisms can metabolize glucose or produce acid or whatever, if they are aerobic or anaerobic, to a variety of properties, metabolic properties, that can be listed in a table, and then you make a classification based on present absence of a given function. But these classifications were artificial and not based in phylogeny, in phylogenetic relationships. Things change a lot. From the 60s, from 1965 indeed, when molecular phylogeny was born. And this was based in a pioneer idea by Emil Zucker-Kandel and Linus Pauling, who proposed that in the sequence of macromolecules, of biological macromolecules such as DNA or RNA and proteins. It means in the sequence of nucleotides in nucleic acids and in the sequence of amino acids in proteins, evolutionary information can be stored. And that it appears to be a simple idea that has a huge and immense success afterwards. And it is the basis of today comparative genomics. All the genomic stuff that you want in the world is based in this particular idea. And then starting from this idea, it would be possible, in principle, to take one molecule that is present in all kinds of organisms because, let's say, it is an essential function, something that works in all cells, and then using the sequence of that particular molecule, build universal phylogenetic trees, means where organisms are placed as monophyletic groups if they are evolutionary related, like making genealogies of organisms. And there was someone at the time that had the idea of applying this particular principle using as universal marker for all organisms the RNA of the small subunit of ribosomes, because ribosomes are essential for all kinds of cells. It is the place where protein synthesis takes place. And so this RNA molecule is essential. There are other RNAs in the ribosome, but these appear to have the right size at the time. So you could use, in principle, this molecule because it is present in all organisms, and because it is essential, it is also a conserved molecule. It means there are some regions that are really necessary and you cannot change them because you lose the function. But it is also relatively variable. So it means that you can compare differences. You have regions that are conserved that you can align, and you have regions that are different that can inform you. In that place, in the variable regions, you can recall or record evolutionary differences that accumulate with time. And that is the idea below or underlying all the comparative genomics and phylogenomics that we do today. So you could use, in principle, such a molecule as you could use a bone to reconstruct the evolutionary history of vertebrates along the fossil record. Except that for microbes, we don't really have a very good fossil record, but there is a fossil record in the molecules, in the DNA sequence. So this is the type of, this is an analogy, but it is quite true, that you can recover this information that is somehow hidden within the sequence of the molecule. So by using this idea, and in particular this molecular marker, Carl Woese achieved what is called by some people a true revolution in biology, and I think it's partially true. He applied this idea of comparing all organisms going from animals and plants and fungi here to classical bacteria, and even to bacteria that lived in very weird environments, extreme environments, as we will see later. And he achieved two different things. One is, yes, he could reconstruct a universal tree of organisms based on the sequence of one particular molecular marker and place together in a tree animals and plants that go together, form a monophyletic cluster with bacteria, for instance. So all organisms can be compared using the same character. And the second thing that he achieved was a kind of side effect, because by doing so, he recognized that a group of weird bacteria, that grew in very weird environments, highly acidic or highly hypersaline or very hot environments, he discovered that this group of prokaryotes form a group apart from bacteria. And he called this group the Archaebacteria originally, and later he renamed this group as the Archaea. And this is the classification that we follow today. We recognize three domains of cellular organisms: the bacteria, the Archaea, and the eukaryotes. Okay? These molecular techniques allowed a kind of a second revolution a bit later, in the, let's say, late 80s, 90s, and we are still in this revolution today. And the idea there was the following. Microbiologists knew for a very long time that only a very tiny fraction of the microbes that are present in the environment, can be whatever kind of samples, only a tiny fraction, usually less than 1%, but it can be 0 or it can be 10, depending on the effort you put into that, can be really cultured in the lab. So this means that most biological diversity, most microbial diversity, escapes cultivation in the lab, and then you cannot study these species and learn about their properties, etc. You cannot have access to these organisms. They live in nature, maybe because you cannot cultivate them because we don't know exactly which are their requirements. And in many cases, you cannot cultivate them because they cannot live together alone. They interact with other species, and then they depend on other species to live. So that explains part of our failure to isolate pure cultures from microbial species in nature. So one potential strategy to sidestep this problem is to extract DNA from the whole community, so all the genomes, all the collection of genes are mixed together, and then amplify by a technique that we call PCR, polymerase chain reaction. You can amplify a particular gene, so a marker gene such as the ribosomal RNA gene, and then in this way you get a collection of molecules. There are molecular markers for all the microbes present here. So by sequencing these different molecules that you can amplify, so you can separate them, etc., with variety of techniques, you can place the sequences in a phylogenetic tree and classify these organisms by establishing phylogenetic relationships in a tree of reference. So, and this is the way that we explore today most environmental communities. So we isolate DNA, we amplify gene markers, so we know by the identity card of the different organisms who is there in the sample without cultivating these organisms, because we cannot. And we do even more things today. I will give you some examples. We not only amplify one single gene, but we can study the whole genomes of a community, this is called metagenomics, and we can even work with genomes from single cells. I'll mention this a bit later. So there are a variety of molecular techniques, but the principle underlying all these methods that we use to study the gut microbiome, etc., all are based on the same idea. There is phylogenetic information contained in some molecules that are conserved, and you can place in a tree, and this gives you information about the group they belong to. So applying these techniques, these environmental microbiology techniques, to many different types of environments on Earth, going from more traditional environments such as oceans or freshwater systems or forest soils, but also from extreme environments that we will see later, such as hypersaline system, deep sea vents, or very cold environments. We have realized that life is much more diverse than we ever thought, and that most of this diversity is microbial. So this is an updated phylogenetic tree of life based on ribosomal RNA genes, and having today many millions of sequences of this particular marker, because we have really millions of sequences now. So each different triangle here corresponds ideally to many thousands of sequences belonging to a particular group. So what we have done with these many million sequences is that validated this tripartite division of life in three major domains: the bacteria and the archaea, and then the eukaryotes. Bacteria and archaea are prokaryotic organisms that have a simpler cell structure in principle, but they are extraordinary. They have a wide variety of metabolisms, especially regarding the way they obtain energy and they fix carbon or gain carbon, much wider than eukaryotes. Okay, so most metabolical diversity is found here. Eukaryotes are more restricted in those terms. And then we have the eukaryotes, which in this tree appear as a third domain linked to this particular point. And now we know that eukaryotes, there is increasing evidence that eukaryotes form a third domain that is not primary, that is derived from the symbiosis of archaea and bacteria. But this is not really depicted here. This is based on a gene tree, so we have these three domains. So it means that eukaryotes likely evolve for sure later, deriving from interactions between archaea and bacteria. So this third domain, the eukaryotes, is composed of organisms that are multicellular, animals and plants that we know very well. And here you have some stars indicating the groups where multicellularity has appeared. And by multicellularity, I don't mean colonies, because colonies, bacteria and archaea know how to do that as well, but really differentiated tissues with certain complexity. And there are two things about that. One is that because of the because these stars are dispersed in this schematic tree, this implies that multicellularity has evolved independently several times during eukaryotic evolution. So that's something very easy to do. It's nothing complicated. It has evolved many times with different genes involved into that, but it's not a big deal. So that's one example. And the other important thing is that these groups where multicellularity has evolved are just relatively minor groups, or minor. Well, they may be quite diverse. Animals are highly diverse, and plants as well. But genetically, if you compare with the other eukaryotes, they are just another group. So this means that most diversity, even within the eukaryotic domain, corresponds or belongs to microorganisms, unicellular eukaryotes. So these are the most genetically diverse within the eukaryotes as well. But we have the same problems to study them. They are small, and you cannot identify them that easily. So that's very important for you to have into account. This is a whole a broad picture, a tree of life picture of biological diversity. And I'm not going to talk about viruses, but these organisms of the three domains of life have all their load of parasites, molecular parasites. Viruses are molecular parasites, highly specialized, and each one of these domains has this type of parasites, as they have also transposons, plasmids, and all kinds of other molecular parasites. Okay. So that's for a general comment about that. So, but there is a problem here. You will notice that there are many triangles here that are darker. So this means that by using precisely these methods or these molecular techniques that allow you to study microorganisms in nature, even if you cannot cultivate them in the lab, what happens here is that you can identify groups of sequences that correspond to organisms existing in nature for which you don't have culture microbes or culture representatives in the lab. So you recognize, you identify groups that can be very broad and have a lot of diversity, but you have no idea of what these organisms are doing in the environment because you cannot study them in the lab. So how to study these organisms, how to explore their functions, for instance, or their ecology, what are they doing in nature? So there are a variety of methods or approaches that microbiologists use to try to study precisely these, the functions of these organisms that are uncultured, that remain uncultured. And one of these techniques is, well, many of these techniques are observational. You have here one example, fluorescent in situ hybridization. You can see these organisms. You can identify them in mixed, in natural samples by hybridizing short oligonucleotide probes that you can label with a fluorochrome, for instance, and you can hybridize because you know the sequence, the original sequence, you have get it from these particular methods. You can design specific probes that hybridize against a particular type of microbe that you have never seen. And then you can hybridize this probe with natural communities and light individual cells in complex mixtures. So you can count them and you can see whether they have particular morphologies, whether they are associated with other organisms or not. So this is fluorescent in situ hybridization, fish, so you can fish them out. And you can also label various probes with different colors and then try to identify different partners in a given complex community. So this is a technique that is quite used. Then you can combine other type of complementary techniques to try to identify the function of uncultured microbes. And I'm giving you here just one example. That is the discovery of a new form of metabolism that was not known of until the early 2000s, anaerobic methane oxidation. a form of metabolism that was supposed to be too low energy productive to sustain life, but that in reality it exists, and it exists because it results from the symbiotic interaction between two different types of microbes. Archaea, a particular kind of archaea that carry out methanogenesis in reverse. They are methanotrophs. They oxidize methane, and these archaea are here shown in red with a particular probe by fish, and then sulfate-reducing bacteria labeled here in green that are associated with this archaea. So there is interspecies hydrogen transfer between the two, so that the whole metabolism, the whole metabolic procedure the process can proceed. And in addition, you can do other things like identifying the kind of lipids from sediments. For instance, this type of consortia develop in deep sea sediments in many anoxic areas, notably at the transition between the sulfate and methane areas. And then you can even make other type of studies and study the fractionation of carbon delta 13, so the fractionation of carbon, to explore where they sit, and then you can identify in this way that these organisms have have a very strong signature of delta 13 with very highly negative values, up to -100, and this is a signature for this type of metabolism, okay? Anaerobic methane oxidation. Anaerobic and methanogenesis is around -60, etc. So we are very far from signatures from, let's say, photosynthesis, as we heard this morning, around -20, 25, something like that. So this is a signature for this type of metabolism, but there can be interferences with hydrothermal activity, so it's not that clear to analyze that type of fractionation. But here we can, and we can do that at nanoscale on particular consortia. So this is one example of how, combining different techniques, you can analyze, study metabolisms and ecological functions in natural communities, sidestepping culture. You cannot cultivate these organisms. And the other way that is exploding these days is to go and study the genomes, to mine genomes for genes encoding particular proteins that you can associate to a given function. And so one of these big types of approaches is called environmental genomics or metagenomics, whereby what you do is you extract the DNA from a given community. It can be plankton, for instance, and then you can you have two ways. One, you can sequence massively with new sequencing techniques all the DNA from a community. It could be the Amazonian forest, but well, you can go to the Amazonian forest and count the number of trees, etc. But it is highly used for microbial communities. So all these studies about the human gut, microbiome, etc., that they use this type of approaches. So you can sequence the DNA of a community, and then in this way you can have access to the gene content of a given environment. So you can associate genes and functions to particular environments, and if you are lucky and you have a particular organism that is very abundant in a community, you can even reconstruct genomes for particularly dominant organisms. But in most cases, you will be able just to assign genes to a community and not necessarily to just one organism. And the other way that to go from this community DNA is to construct what people call metagenomic libraries, where you can clone big pieces of DNA, so not the whole genome, but big pieces of genomes, and then you can mine, you can look for particular clones and study in this way not only the gene content, but because you have big pieces, and normally all the genes in the same piece of DNA belong to the same organism, then you can see if there are things together. And if you have genes that are highly conserved, you can place those genes in a tree, in a phylogenetic tree, and then all the genes that are around in the same, sitting in the same on the same fragment, they will belong to these organisms that you can now place in a phylogenetic tree. So you can assign functions to particular organisms by using this type of approach here. And there is another approach that is complementary and that is also developing very importantly nowadays. That is single-cell genomics. And here what you do is to, instead of sequencing massively the whole mixture, sometimes microbial communities on Earth are, in most cases, except in extreme environments, they are extremely diverse. You find thousands of, let's say, microbial species. I quote unquote, because microbiologists don't like to speak about species. That's a very difficult thing to define. But anyway, some kind of operational taxonomic unit. So you can define these taxonomic units, and then what you can do is, one thing that you can do is to try to isolate. Instead of doing that, you can try to isolate cells by using particular techniques, cell sorting or micromanipulation in a complex community. And then you can try to isolate parts of information from that mixed community that can be highly complex, as I say, thousands of operational taxonomic units. Here you can go one by one to study those genomes in a community, and the two techniques are actually quite complementary. So you can amplify the genome from single cells in principle, even if there are some biases, etc., and it's not an easy task, and then obtain this type of information that you can contrast with the rest of the community. So this is how we microbiologists study microbes in the environment, and this is providing a wealth of information. And among this type of information, at the beginning is something very descriptive. What we find is a high diversity, and even, I don't know, each new study brings new groups that are being detected in this way. This is just one example. So some people call about the microbial dark matter, so those lineages for which we don't have that much information, but we know we are able to detect them in the environment. So this is just one example, Nature 2013, where looking for single cells or isolating single cells from different environments by these methods that allow you to sort individual cells. So you put one cell in per tube, and then you can amplify the genome, and then you can sequence it and get draft genomes or partial genomes in general from these cells. Then you can detect in this way, these people have been able to identify or generate partial genomes for groups of organisms that had never been detected before. So the variety, this is just a schematic phylogenetic tree, the variety of lineages for which we have now partial genomes, but we have no idea of what these organisms are really doing in nature, it's really important. So this is a kind of a summary of how you can assign functions to uncultured organisms. So this is just to, let me see what time it is. I can follow a little bit. This is, you tell me when we stop for coffee. So this is just a summary to say that we can either isolate and cultivate these organisms in the lab, study the phenotypic properties, the genomes. Then we can have interdisciplinary approaches combining a variety of techniques to study microbial functions in situ, in the natural environments. And then we can use these genomic or metagenomic approaches to mine genes and their functions in natural communities. So a combination of the three is really what is today the way to go and learn more about microbial communities. So coming now back, and this is in part how, coming now back to this universal tree of life, if you want, with the three domains. I have to say that when the Archaea, I'm going to focus particularly on the Archaea, just to say that when the Archaea were discovered, they were discovered in the late 70s. At the beginning, well, people were reluctant to say they were a different domain because they really look like bacteria phenotypically. So we have learned about that. But then, because some of these Archaea were able to grow in the lab, people started to study their molecular biology. And by studying their molecular biology, we quickly recognize that even if their phenotypic properties appear like those of bacteria, their molecular biology is really eukaryotic-like. This means that all the informational processes, so the proteins involved in replication, in transcription, in translation, among others, are much more similar to the eukaryotic ones, or have only homologs in eukaryotes but not in Archaea. So actually Archaea are much more eukaryotic-like than bacteria. And today we know that this is likely an issue of vertical descent in a sense, because eukaryotes derive in part from Archaea. And actually many, what happens in general is that many prokaryotic, eukaryotic complexes involved in molecular biology, DNA replication or repair, have many more proteins than the archaeal ones. So paradoxically, we have learned many things about protein-protein interactions and specific roles of proteins in eukaryotes by studying their archaeal homologs, because the complexes are far simpler, so they have less subunits, for instance, and so it is easier to pinpoint the function of an archaeal protein, and then by analogy try this function in the eukaryotic homolog. So in this way, Archaea have been very useful to learn more about eukaryotic molecular biology. That is for the anecdote. And the other important point about the Archaea, and this is an introduction to the second part of my talk, is that when they were discovered, they were essentially isolated, those that were known at the time, from very weird environments, very what we call today extreme environments, very hot environments, or also very acidic environments, very salty environments. So in a sense, Archaea were conceived at the beginning as a domain of extremophiles. We now know this is not the case, because many Archaea thrive in normal let's say more normal environments. You find them in the oceans, you find them in soils, you find them in animal guts. You have many methanogens, and so they are common in other environments. But it is always true that most extremophily records belong within the Archaea, so that they really cope with extremes, physicochemical streams, that other organisms cannot. So that's what brings me to the second part of my talk, that deals with life in extreme environments. And I don't know, you tell me really when I have to stop. I can go for a while, no?
Yes, in half an hour.
Okay, perfect. So, good. So life in extreme environments. Second part of my talk. Actually, life in extreme environments, when you look to this type of environments, and for a very long time, this type of environments were thought as of sterile environments, places where life could not occur, simply because there is no macroscopic life. So you don't see plants or animals, and then for a very long time, biologists were not interested in this type of places. But in the perhaps one of the earliest records of this type of organisms date back to the 1930s, where when Eleazari Volcani isolated for some organisms that were adapted to high salt extremes, to very high salt concentrations in the Dead Sea. So showing that the Dead Sea is not dead, but there is life in the Dead Sea. So these were organisms that could grow at very high salt concentration. And by the way, it's not only that the salt concentration is very high, it is that there is a lot of magnesium as well, and this is a bit deleterious for many organisms. So a really highly extreme environment. But possibly the interest in extremophiles raised in the late 70s and 70s, and in the late 60s and the 1970s, when Thomas Brock, famous microbiologist, and there is a very good book of general microbiology that is called, from his name, the Brock's Biology of Microorganisms. And so he started to isolate thermophilic or heat-adapted organisms from different places in Yellowstone. And in 1969, he isolated one organism which is a bacterium, Thermus aquaticus, whose optimal temperature, growth temperature, is around 70 degrees. And then a couple of years later, he isolated an even more thermophilic organism, Sulfolobus acidocaltarius, which is a thermoacidophilic organism growing up to 90 degrees Celsius and at very low pH between 1 and 5. And it turned out later that this organism, thought to be a bacterium at the beginning, was in reality an archaeon, so one of these early members of the third domain of life, the Archaea. So, what is an extremophile? How to define an extremophilic organism? And there are many possible definitions, but in the framework of the extent of life and the limits of life on Earth, we can define an extremophilic organisms or extremophilic organisms in general as group of organisms that for a given parameter, here temperature, develop at the limits of the distribution of this parameter within which life can occur. So in this kind of graph where you see growth temperature plotted against growth rate, you have for any organism you have an optimum of temperature, and then if it's too hot you die, and if it's too cold as well. Well, within this type of framework we define more or less arbitrarily, or there's some kind of consensus, different organisms that we call psychrophiles if they love low temperatures, then mesophiles, like we are mesophiles, so our type of, so this is a sign of again anthropocentrism in a sense, because we place as mesophiles, so we are the norm. Maybe it's not the case, or it was not the case in the past, we don't know, but well, this is the way it is. So we compare the rest of the world with us. Then we have moderate thermophiles, extreme thermophiles, and then hypothermophiles growing at very high optimal temperature. So an extremophile would be really an organism living or developing its full cell cycle at these extremes of the distribution for any given physicochemical parameter. And so you can classify extremophiles according to the type of physicochemical parameter that they are facing to. So in general, if you consider temperature, you have organisms that are hypothermophiles. Sometimes people include thermophiles as well. So hypothermophiles are defined as organisms growing optimally at more than 80 degrees Celsius, thermophiles somewhere between 60 and 80. Then you have psychrophiles on the other extreme, growing optimally at less than, let's say, 10-15 degrees. Then you have the pH, and for the pH you can define two groups, the acidophiles living at optimal pH below 2 or 3, alkaliphiles on the opposite living optimally at pH higher than 9 or 10. Salinity, you define organisms that are halophiles at very high salt concentrations up to saturation, equivalent to sodium chloride. Pressure, you define organisms as barophiles or piezophiles, living at very high pressures. And then you have other features that are not necessarily organisms that really depend on these conditions to live. This is the case for true extremophiles. These organisms depend on high temperature, on salinity, on high pressure to live. But then there are some organisms that tolerate some extreme conditions. So you can have organisms that tolerate desiccation and that prefer to be in environments where you have not a lot of water, so hydric activity, so that tend to tolerate anhydrobiosis, and organisms that tolerate important levels of radiation, including ionizing radiation, which you call radiotolerant. And then organisms that tolerate important concentrations of metals, including heavy metals, which can be very deleterious for many other organisms, and that you call metallotolerant. So why are hypothermophiles or other kind of extremophiles interesting? And this is something that they were very these extremophiles, they are always very appealing, etc. But there was really a very important peak of research on extremophiles in the let's say 1990s, because they fascinated biologists at the time. It was the discovery that these kind of organisms could exist, and people were in particularly amazed about the type of molecular adaptations that these organisms could develop, because in some cases, like very high temperatures, this is something that is deleterious for proteins in vitro, and then how these organisms can live at temperatures higher than 100 degrees. So that was something that people was really amazed about, and so this is one of the reasons to know more about their molecular adaptations. There was also an important interest in the potential application of these organisms or the products, so the enzymes, etc., for biotechnology issues, so biotechnological applications. Then there was also a very strong link with evolution because of two reasons. First, because the discovery of extremophiles was tightly linked to the discovery of Archaea, the third domain of life. And second, because this gave rise in parallel to models on the origin of life, which made of hypothermophilic organisms the first organisms ever been on Earth. So there was a strong link. Before we recognized the existence of, in particular, hypothermophiles, there were no hypotheses on a hot origin of life anywhere. So life was only more mesophilic, let's say. And finally, of course, the study of extremophiles is important within the perspective of astrobiology and the potential for life in the universe, because, of course, you can now explore environments that, in principle, would seem sterile, but in reality, now, as we know now, they are not. And I have put here some features of extreme environments that can be generalized to all of them. There is in general a decrease of diversity. This means that you have less different species, whatever species it is, even if you can have very high amounts of biomass of a particular organisms that are adapted to these particular conditions. But the diversity, the genetic diversity, the microbial diversity in this type of environments decreases with when you reach the limits of life. Then there is, of course, a predominance of microbial life, even if in some environments you can have also animals and plants. For instance, in very cold environments, you can find sometimes animals that are very well adapted to very cold temperatures. these are environments where microbe-mineral interactions are more evident or more tight in the sense that well, the natural environment is mostly mineral, and many of these environments, or many of these many of these environments are extreme for more than one parameter. This means that many organisms are facing similar or several extreme parameters at a time. So this is just one example of environments where you find or you may find several extreme conditions at the same time. So you have the deep sea, where you will find barophilic organisms adapted to very high pressure, going down to the Mariana Trench, for instance, 18,000 meters deep. And at the same time, they will be Xerophiles because the deep ocean is cold, around 2 or 3 degrees on average. But they can be also hyperthermophiles at the level of deep sea vents. They can be halophiles in deep sea brines, like in the Mediterranean. You have many of these deep sea brines, and many of these are anaerobes. Although the absence of oxygen is not, truly speaking, an extreme condition. Okay, there are many organisms living, including our gut, without oxygen. That's not a problem. There's a wide diversity of organisms living without oxygen. If anything, oxygen is an extreme condition. Historically, oxygen has been an extreme condition because you have to face a lot of oxidative radicals and free radicals that can be really harmful. There is a variety of adaptations that we have, we extremophiles that respire oxygen to cope against those deleterious effects. Then you have, for instance, the deep subsurface. You have similarly barophiles, but also adapted to many extreme conditions, and on others. And among these others, this is just one example of one very highly extreme place for a variety of reasons. And this is, we made a field trip recently, a couple of weeks ago, to this region, the Dalol in Ethiopia. This is a place at the level of the Danakil Depression, so it's under the water level because well, there was the sea and then it evaporated. And so here at this place, you can face very high temperatures, up to, we measure 115 degrees. Extremely salty, so we are always at salt saturation because the sea, there was an arm of the Red Sea that evaporated. It was some tectonic activity that closed the area, and then it is a very hot place. So you have a very intense evaporation. So there are around 2 kilometers of evaporites that are traversed by this hydrothermal activity. And so everything is, so you have halide saturation, so sodium chloride. So these are chimneys of salt chimneys, actually. And at the same time, you have very low pH. And so we have measured pHs that go down to minus 1.5 and even minus 1.7. So really, the combination of these parameters is quite unique. So this is one of these examples of multi or pluri-extremophilic environments where you, and I don't know what we will find here, but we will study that. So, and now what I'm going to do, if we have the time, or okay, is to simply walk around these different kinds of organisms that are adapted to to highly extreme conditions, and I will tell things about these organisms. So starting by the hyperthermophiles, these are organisms that are adapted to very high temperatures, and they are defined as organisms that develop or have an optimal temperature higher than 80 degrees Celsius. And this is perhaps one of the cases where you see best this characteristic of extreme environments that I was mentioning before, the fact that you have less diversity when you go to the very limit of that parameter, that physicochemical parameter. Because if you place this kind of thermometer of life, you will see that at relatively mild temperatures, let's say between 0 and 50, 60 degrees, you have a variety of things. You have all kinds of organisms belonging to the 3 domains of life. So belonging to archaea, bacteria, eukaryotes, but eukaryotes cannot cope with temperatures higher than, let's say, 60, 64 degrees. So the most hemophilic eukaryotes that we know are certain fungi that can grow up to 60, 61, maybe 62 degrees, but nothing else. You have organisms that grow at 50 degrees, some thermophilic algae, some ciliates, but most eukaryotes are adapted to lower temperatures. And below or above 60, 65 degrees, you have only prokaryotic organisms, bacteria and archaea. But again, when we reach temperatures of around 90, 95 degrees, those are the upper possible temperatures for bacterial growth. Beyond that, we only have archaea. So the most hemophilic bacteria, and there are not many, there are 2 genera that are very well known, Aquifexa and Thermotoga. They can live at a maximum growth temperature of 90 or 95 at the maximum. But really beyond that, we have only archaea. So this means that only archaea can grow at temperatures, optimal temperatures of 100 degrees and above. So you see very well this decrease in diversity, including at a domain level. So what kind of environments? In continental areas, you have this type of hot springs, for instance, or mud volcanoes in some volcanic areas. In Yellowstone, for instance, so these were among the first hot environments studied. And you have a picture here of Wolfram Zillig, one of these extremophile and particularly hypothermophile hunters. So he and Carl Stetter were pioneers in isolating many archaea, specifically hypothermophilic archaea, from these areas. And so many of these sulfolobus, etc., that are now in culture collections come from these people. But you have also this type of hypothermophilic organisms living in deep sea vents, where you have very interesting communities that form a kind of hotspots of biodiversity in the deep sea. The deep sea is largely, it's not a desert, but almost so, in the sense that it's, especially the deep water, is highly oligotrophic, poor in nutrients. But in this type of systems along oceanic ridges, you have communities of organisms, including animals, that are highly diverse. And this is because you have here around this area the confluence. This is not only because of hypothermophiles, but they develop there, but also because you have the confluence of a reduced and an oxidized world at the same time. So in these systems, what you have is the seawater that percolates and penetrates within the upper layers of the mantle. And then if you have hot points that are closer in oceanic ridges, for instance, then you have this water that can solubilize metals and that can enrich in reduced gases like hydrogen sulfide and methane and hydrogen sometimes. And then you have these fluids that are very hot, can be up to 350 or 400 degrees, too hot for life, in chimneys that we call black smokers, but also in colder vents that can be white vents at lower temperatures. And here you have all these gases that are highly reduced, and then the seawater, which is highly oxygenated. So you have at the same time a variety of electron donors and electron acceptors, mostly oxygen, but you have also sulfate and nitrate. So you have a variety of possible redox reactions that are the energy source for many different types of organisms that are chemolitho-autotrophic. That means that gain the energy from this type of redox reactions, and they can fix carbon at the same time. So we talk about these systems as chemosynthetic ecosystems, because they are not necessarily based on light. Indirectly they are, because most oxygen comes from photosynthesis anyway, even in the deep ocean. But in general, they do not depend directly on light to set up a trophic chain, which in surface waters you do with light. So these environments are in general hotspots of biodiversity. Coming back to hypothermophiles, these are the places where the most hypothermophilic organisms have been isolated, because one thing that is essential for life is the presence of liquid water. And so you cannot have life at 100 degrees at the sea level, but you can have it here because of the pressure. So you have organisms that can live at optimal temperatures higher than 100 degrees. And for a long time, the record of hypothermophily was held by Pyrolobus fumari, which was isolated from the mid-Atlantic ridge, and which could grow up to a maximum temperature of 113 degrees. And then there was another strain that was published as saying, okay, maybe this organism can grow up to 121 degrees. Maybe yes, maybe not. But the fact is that when you are a microbiologist and you're working with this type of microbes, you have to deposit, if you describe a new species, you have to deposit this organism in a culture collection so that your colleagues can validate and see whether this finding is true or not. And this strain was never deposited in a culture collection, and it was lost. So we don't know if it was really growing until 121 or not. But we know now that there is one strain of Methanopyrus candleri. This is one archaeon, a methanogenic archaeon producing methane, that can grow, a particular strain of this organism, that can grow up to 116 degrees at atmospheric pressure. But if you put it at a pressure that is normal at the depth where it lives, it can grow up to 122 degrees. And this is in general, it's a common observation. When you put organisms that you isolate from these depths at the pressure, the local pressure, they extend their optimum and their maximal growth temperatures a bit. So this is so far the most hypothermophilic organism known, so for the record. I mentioned before that each domain of life has its cohort of viruses and other kind of molecular parasites. This is true also for hypothermophiles, and you have many different families of viruses that are specific of this type of organisms, in particular of Archaea, hypothermophilic Archaea, sometimes with many funny shapes, etc. So just to mention that there are also a variety of parasites that are associated to these very particular organisms, and therefore they can cope also with this type of temperatures because they're host to. Hypothermophiles are also important for biotechnology, or at the beginning, because many of their enzymes, of course, can be purified and utilized for industrial processes, for instance, to bioremediate or industrial waste, for instance, or you can do bioleaching at high temperature. You can also use some of these enzymes, proteases, lipases, as additive to the detergents to wash your clothes at high temperature. But perhaps one of the most important applications of thermophilic or thermostable enzymes is the PCR, the polymerase chain reaction, because there is based in the amplification of DNA using one DNA polymerase that is thermostable. These polymerases can come from a variety of different thermophilic organisms, and this means that these polymerases they amplify DNA at a given temperature, that the famous TAC from Thermus aquaticus amplifies DNA at 72 degrees, but they can tolerate much higher temperatures, and then they can tolerate denaturing temperatures. So you can make cycles where you amplify DNA by using this polymerase, and then the newly formed DNA duplexes, you can denature them at much higher temperature, at 95 degrees Celsius, for instance, and the polymerase is not acting at that temperature, but it can resist. So when you go down again to 70 degrees, it starts acting again. So this property is exploited for this polymerase chain reaction that is used widely in all molecular biology labs, in the police labs. So when you have some murder and then there is some hair or a bit fragment of skin, this is the technique that is used, and so it is thanks to thermophiles and hypothermophiles that we can use it so widespreadly. And okay, this is just a table for you to see that it's not a joke. So there are many commercial enzymes that are thermostable, and so here you have a list of these. It's not very important that you read this, but this is just for you to know that this is happening. And then about the molecular adaptations, hyperthermophiles are very special because they have to cope, and this has fascinated microbiologists and in general biologists for many years, because they have to cope with potentially highly deleterious effects. So when you raise temperature, you can of course denature proteins, that is a problem, or you can degrade nucleic acids, for instance, by deamination of cytosines, and you can denature simply nucleic acids, for instance, in particular nucleic acids that are not circular, and that then can be denatured, and that can be really deleterious. But they can also, high temperature has another highly deleterious effect, that it is that it increases membrane fluidity. And this is particularly dangerous for cells because of proton leakage. As I mentioned before, cells make an energy budget from the creation of a proton gradient across membranes. So you need, in order to be able to generate that gradient and to get energy from that gradient, to maintain it. And if you have very high temperatures, you will have proton leakage. It means that the membrane will become permeable to many metabolites, including in particular small ions. And then it is all the energetics from the cell that is disrupted in this way. So in order to cope with these deleterious effects, there is a number of many adaptations. At the level of DNA, to cope with degradation and denaturation, you have, in particular for hypothermophiles, so organisms living at very high temperatures, a particular enzyme that is a marker for hypothermophily, although there's also some thermophilic organisms living at lower temperature that also have a reverse gyrase, but all hypothermophiles do have one. It's an enzyme called reverse gyrase. Reverse gyrase, what it does is that it introduces positive supercoils in DNA. So temperature has this tendency to unwind the DNA duplex, the double helix, and then this enzyme will introduce positive superturns, so that in the end the geometry of the DNA molecule will stay at a level that is comparable to our own DNA geometry, for instance. So actually, it counteracts the effect of the unwinding effect of temperature. At the level of RNA, we see in the case of structural RNAs an increase in, and then for the DNA there are also, but this is also true for RNA, protective DNA binding proteins that can protect DNA from thermodegradation and also very efficient repair mechanisms. In the case of structural RNAs, but not of messenger RNAs, I think that for messenger RNAs, what happens is that there is very high turnover. But in the case of structural RNAs, such as some of the ribosomal RNAs, what we observe is an increase in the GC content just because you establish more hydrogen pairs than when you have simply AT. And this increases the internal cohesivity, the cohesion of molecules, and then they can they prevent in this way denaturation. At the level of the cytoplasm, we observed in general a high salt concentration or also compatible solutes. In general, the viscosity of the cytoplasm is increased. The adaptation of proteins, in general, you have more compact proteins with more internal intramolecular interactions, just to keep the three-dimensional structure of proteins and also their activity. So we observed in general a high hydrophobicity to increase hydrophobic interactions within the molecule. Sometimes you also observe more charged amino acids because you can establish salt bridges. Well, as I said, more intramolecular interactions, and you also observe the reduction of loops. So many loops are lost, and side chain reduction to avoid an increase of flexibility. And there's a variety of chaperones and heat shock proteins that help proteins fold correctly. And finally, at the level of the membranes, and the problem here is proton leakage, you have in general that archaeal membranes, and remember, archaea are the ones that are really well adapted to hyperthermophily, are naturally more fit or better adapted to high temperatures because they are less prone to proton leakage, in part because they have, all phospholipids are made of glycerol phosphate, and then lateral chains that in the case of archaea are as isoprenoid chains. In the case of bacteria are in general fatty acids. The difference, one of the difference between archaeal and bacterial lipids is that archaeal lipids join this glycerol phosphate to the lateral chains with an ether bond that is more resistant than the ester bond that bacteria use. So ether bonds are really apparently one adaptation to these higher temperatures, and also instead of being bilayers, so a double layer of phospholipids that are placed one in front of another, what you have is that these lateral chains, they fuse, forming monolayer lipids, so lipids that traverse the membrane. So this increases the rigidity of the membrane and prevents proton leakage. So this is one potential adaptation. And actually many bacterial, so the lipids of bacteria that are able also to cope with high temperatures mimic adaptations that are present in archaea. As I said, bacterial lipids use ester bonds to link the fatty acid chains to glycerol phosphate, but these bacteria, they use ether bonds instead of ester bonds. And in many cases, you also see the fusion of these lateral chains to have transmembrane phospholipids. So these are particular adaptations. And I think that now we stop for the coffee break, right? And we come back, I will just mention something about possibility of a hypothermophilic Luca. Okay, I stop here for the coffee break.